The first speaker is Erika.
Thanks for all for coming back from a New York lunch, and thanks Francesca and Frank for this fantastic meeting. The only regret I have is it's not twice as long, because I know half the audience has fantastic stories that we're not hearing about. So next time, make it 2 days. That would be the only way to make it better. So I'm going to talk today about motors and the importance of axonal transport. And fortunately for me, all the major concepts I'm going to talk about have been introduced. That allows me to put in 2 news stories that we're working on, which is exactly one too many, but I'll go as fast as I can and try to distract the timekeeper. So we already heard about the problem and fascination of axons, how you have these long distances that have to be traversed by molecular motors, kinesins and dyneins, moving along microtubules, which is something that Francesca and I are equally obsessed about. So you could study any of the organelles that move along the axon and you'd have a great story. They've all been really interesting for us and others in the room. Today I'm going to talk about 2 stories related to the trafficking of signaling endosomes and synaptic vesicle precursors. And I chose these because they illustrate complementary ideas about axonal transport. One is high-speed express trafficking, the type of trafficking you need for signaling endosomes, and the other is very specific local delivery, and that relates to synaptic vesicle precursors and their delivery to synapses. So first, to talk about signaling endosomes. We know that this trafficking is driven by dynein, but this brings up a really interesting question. Dynein is the only major minus end-directed motor in neurons, so we know that it has a wide variety of cargoes that it has to traffic. And this has been a mystery, how you could get very cargo-specific regulation. So we know this is important, and this is a neurodegenerative meeting, so I'll point it out. We know that since dynein drives so many cargoes, we know that mutations in dynein, the interacting complex dynactin, and effectors such as BIC-D, and I'll tell you a little bit more about dynein effectors in a minute, are all involved in a range of neurodevelopmental and neurodegenerative diseases. So that brings us back to this question of how we could get organelle-specific regulation of the motor in each context. Well, fortunately, new insights from a number of labs came out in the last few years suggesting a model that might apply. First of all, we have a dynein that can be auto-inhibited in this phi complex, which becomes opened up and then interacts with dynactin. But then you have interaction with an effector that forms the active and highly processive motor complex. We know from beautiful images from Andrew Carter that the effectors, in this case BIC-D, bind along the groove between the ARP1 filament of dynactin and the base of dynein. So basically threading through this structure with a long extended coil. That forms a very stable complex that can then be highly processive. So this is true for BIC-D, which has been implicated in the movement of Rab6 vesicles as well as RNA, but it's remained unclear what are the specific effectors, dynein activators, for other different organelles. So we set out to ask, we and many other labs, if there's a toolkit of dynein-specific effectors that could activate each of these organelles. In particular, we focused several years ago on members of the hook family. We did this because of beautiful work in model organisms showing that in filamentous fungi, loss of the hook homologue, either hook A or hook 1, specifically disrupts early endosome transport. So we reasoned fungi are a great model system for neurons, believe it or not. We reasoned that maybe these proteins would be important in trafficking invertebrate neurons. So here's the overall structure of the hook proteins. They have the extended coils that you see in BIC-D. They have a motif that's been shown in another dynein activator called spindly, but they have a distinct N-terminal called a hook domain. Here's a little bit blurred out picture of what the hook proteins look like when they're dimerized. You see the full extended molecule in this long coil domain. For previous work had suggested that hook 1 was generally implicated in endosome trafficking in mammalian cells, and hook 3 localized to the Golgi, with hook 2 being recruited to centrosomes. So with that, we set out to ask if hooks could activate dynein, We showed in single molecule assays published several years ago, and there's also beautiful work from a number of different labs showing the same thing, that hook proteins, hook 1 and hook 3, can activate dynein even more than BIC-D. So essentially, they make a super processive dynein complex. You get fast velocities and long run lengths. We could also show that these ability to activate dynein critically depended on the extended coiled coil, which was not surprising, but also required this novel hook domain at the end of the hook's molecules. It was originally suggested to be a microtubule binding domain, but this is not true. It's EB1-like, but lacks the ability to bind directly to microtubules. We now know from crystal structures just recently solved with Roberto Dominguez, also at Penn, that this hook domain binds directly to the dynein light intermediate chain, and this binding is required to form that super processive complex. So if we look at chimegraphs of single molecules of labeled hook 1 moving in a dynein-mediated way along microtubules, you see these long processive runs in the control case. You can completely block that motility by blocking the binding here with a helix peptide. If you mutate that helix and the specific residues that interact, you regain the motility. So all of that tells us that yes, vertebrate hooks can specifically activate dynein, but doesn't tell us what organelle it might be moving. And we have quite a few to choose from in the axon, so we decided to dig in and look. Based on previous work, we initially started by looking at Rab5 and Rab7 positive endosomes. And we do see, when you express hook 1, that it does co-localize with both Rab5 and Rab7, but when you quantitate that, it's only a subpopulation of each. More depressingly, when you knock down hook 1, you see really no effect on Rab5, overall Rab5 motility or Rab7 motility. And thanks to our reviewers, we also know you see no effect on the trafficking of mitochondria or autophagosomes. So this starts to sound like a negative story. But fortunately, I have a fantastic student, Mara Olenek, and she said, let me look more closely at these chimegraphs. And what she saw was that the labeled hook 1 co-localized with a population of very fast-moving, highly processive Rab5 positive endosomes. So you can see that the general population of hook 1 positive Rab5 endosomes moves in a more retrograde direction and with faster velocities than the overall population. She also looked at where hook 1 was localized in neurons. Here we see the spread of the soma, mid-axon, and distal axon. You can see, for instance, BIC-D is enriched in the soma, and RILIP you see in the mid-axon, but hook 1 is localized distally. So if you put together what I just said, that you have a distally localized population that's Rab5 and Rab7 positive, then you're thinking signaling endosomes, which we know are generated distally and traffic signals back to the soma to change patterns of gene expression. So we could model this then in hippocampal neurons, looking to see whether hook 1 is required for the motility of either TREC-B or BDNF. I won't show you the TREC-B results. I'll just focus right in on BDNF, which we can attach to quantum dots, which gives us a bright, stable signal to monitor over time. What you can see in the chimegraphs here or the drawings below is these BDNF quantum dots move in these very rapid, highly processive movements from the distal end of the axon back to the soma. However, that trafficking is significantly inhibited if you knock down hook 1. Just to ensure that we're doing a specific knockdown, we can rescue with halo-labeled hook 1, and we can restore the motility. So this was done in a bath application of BDNF, but then Mara applied microfluidic technology where we can specifically add the BDNF quantum dots to the distal terminal of these axons and watch trafficking back to the soma. Under these circumstances, you can see in the mock-treated cells, we see this beautiful trafficking of BDNF-positive quantum dots, whereas in the mock-treated cells, you see a significant inhibition of this long-distance transport. And that's all quantitated here. So if this is true, if you do need hook 1 to mediate this trafficking long distance, the final output is changes in gene expression. So can we see a change in the signaling when we knock down hook 1? So our readout here is going to be an increase in phospho-Krebs in the nucleus. So here's the control case. You see a low amount of nuclear phospho-Krebs initially, but if you add distal BDNF, you see a striking increase in that signal, and that's quantitated here. When we knock down hook 1, what you see is no increase in phospho-Krebs in the nucleus and a complete ablation of this induced signal. So together, this gives us a model in which hook 1 is a specific activator of signaling endosomes. It cannot be compensated for by hook 3, and it's required both for the distal uptake and the long-distance trafficking of signaling endosomes back to the soma to change patterns of gene expression. We're open to the idea that there might be a transition in activators en route, perhaps to RILIP, and we're looking at that right now. So this is a model, as I said, of long-distance, very rapid express transport. But when you're shipping things, you also have to worry about precise local delivery. So for the rest of my talk, I'm going to talk about that. I'm going to pick up some of the concepts that Lily introduced us to and basically echo some of the major findings of her talk. So basically, here we're looking at the question of how you get synaptic vesicle precursors to their site of function, to en passant synapses in hippocampal neurons. So we started this by growing our hippocampal neurons 14 to 18 days in vitro and watching the trafficking of labeled synaptic vesicle precursor proteins. What you're seeing here is our initial labeling with synaptophysin, and then we bleach the signal and then watch it recover over time so that we can watch new trafficking into this region. What you can see from the chymograph is the long anterograde trafficking of this synaptophysin with pauses that are not always but frequently coincident with synaptic sites. We can quantitate this here where we're looking at the delivery of new synthesized material to a collateral, axon collateral, and we can see that the new delivery is almost entirely anterograde and very little is retrograde in nature. That's quantitated over here. We see that this specific anterograde delivery and synaptic retention is specific for a synaptic vesicle marker, but not seen with something that wouldn't be expected to be highly enriched at the synapses, such as LAMP1. We can then take this further and ask, do these vesicles pause preferentially at presynaptic sites? And we can see that they do when they're moving in the anterograde direction. We don't see any significant pausing when they're moving in the retrograde direction. So again, that's what you'd expect, but made us want to think about the underlying mechanism, much as Lily did and introduced us to previously. What I'm going to focus on is the motors that are involved. We know that synaptic vesicle precursors are driven by the motor KIF1A in comparison to conventional kinesin, which primarily is the primary motor for LAMP1 positive vesicles. So we wanted to ask, are there specific signals between the microtubule and the motor that would give us this specific pausing? And if you were here before lunch, you know the answer. You know exactly what it's going to be. And I don't have to show you this, which I think the exact image was shown previously, to remind you about microtubule dynamics. So we asked whether dynamic microtubule plus ends would be enriched at synapses. If we'd heard Lily's talk earlier, we wouldn't have had to do this. But we started, actually we did this last because we're a reviewer, but I'll show it first. There's an antibody that preferentially recognizes GTP-tubulin, and we can see a coincidence of GTP-tubulin, which would be enriched at dynamic microtubule plus ends at synaptic sites. More quantitatively, what we can do is use EB1 dynamics or EB3 dynamics, just as Lily did, to look at microtubule dynamics and their coincidence with presynaptic sites. And what we see, as she just reported, is that these presynaptic sites are hotspots for dynamic microtubules. And that's quantitated here. I think the only difference is we see significance in both the initiation and the termination of EB3 comets, but otherwise exactly the same theme. We could then ask the question, does it matter? Why would you even care if microtubules are dynamic at synaptic sites? And the easiest way to do this is just to simply damp down microtubule dynamics with nicotazole. If you use a high dose, you'll depolymerize microtubules. If you use a low dose, you'll just damp them down. Under these circumstances, we can watch trafficking to sites, add low-dose nicotazole, and 30 minutes later, look at the effects on trafficking. And what we see is that this low-dose nicotazole has no effect on overall trafficking, either of LAMP1 vesicles generally, or in the retrograde direction at all, or non-synaptic trafficking, but it significantly damps down the specific retention of synaptic vesicle precursors at synaptic sites. So this told us that we were on the right track, that dynamic microtubules at synapses really mattered. But we wanted to know why they mattered. What was reading the signal of the dynamic plus ends? And to do that, we turned to in vitro reconstitution assays, and these have already been introduced. So briefly, what we're looking at microtubules dynamically polymerizing off seeds, and we can add our labeled motors in and watch trafficking along these microtubules in vitro. When we do this, we can recapitulate both the GDP-rich microtubule shift and then the dynamic plus ends, which will be enriched in GTP tubulin. When we add in either our KIF1A motor, which is the one on synaptic vesicle precursors, or the kinesin, or the KIF5C motor, which is conventional kinesin, we can see what's already been reported by a number of labs, especially Kristen Verhey, that KIF1A is a highly processive motor. Once it binds to a microtubule, it tends to run almost to the end. That's different than KIF5C, which is also a processive motor, but with much shorter runs, as you can see here. However, when we analyzed this data in detail, we saw something we expected and something we didn't expect. What we did expect is if we look at where the runs terminate, KIF1A tends to terminate reasonably close to the end, within 2 microns of the end. This is less so for KIF5C because of its decreased processivity. What we didn't expect to see is that KIF1A rarely initiates a run within 2 microns of the plus end. And so this is a deselection for binding to the dynamic plus end of the microtubule. And we wanted to know why. So we generated microtubules that were polymerized with GDP and Taxol, so they'll mimic the microtubule lattice along the shaft of the microtubule, as well as microtubules polymerized with the non-hydrolyzable analog GMP-CPP. And we can put these in 2 different colors in the same chamber and then ask, do these motors bind with any preference? When we do that, we see that kinesin 1, either KIF5B and KIF5C, 2 different isoforms, show no preference for binding to either the GTP-like or the GDP-like lattice. However, KIF1A shows a remarkable propensity to bind to the GDP, but not the GTP lattice. We can further show that it's also competed off from the microtubule end by the presence of EB binding proteins at the end. So if we either decorate the microtubule with a high level of EBs, we see reduced binding, or in modal assays, when we're running these KIF1A motors down, we can see a dissociation event when it reaches the EB1 at the end of the microtubule. So for this, we have a model that KIF1A is going to be highly processive, move down the shaft of the microtubule, but once it reaches these dynamic plus ends that are enriched at synapses, it's going to have an increased rate of dissociation, leading to cargo delivery at sites of need. But we wanted to go a little further to test this model. So KIF1A is known to be required for this synaptic vesicle precursor delivery, and it's also known that there are many different mutations in KIF1A that cause both neurodevelopmental and neurodegenerative disease. So we screened through them to find a mutation that was specifically within the KIF1 motor domain and specifically within a loop that's proposed to contact the microtubule, in addition to the actual motor contact. This mutation has previously been suggested to affect ATP-dependent binding to the microtubule. So we asked whether KIF1A with the T258M mutation would be less responsive to the difference in the microtubule lattice. And here in that same assay that I introduced you to before, where we have either the GTP-like or the GDP-like lattice, we can see that this mutant kinesin is no longer capable of discriminating between these two lattice structures. So then we asked, would this, because it lacks this ability, and if we put it into our dynamic microtubule assay, would we have that same ability to detach 2 microns from the end, or would it go all the way to the end? And here from these cumulative distribution frequencies, you can see whereas the wild type is highly likely to detach when it reaches the GTP-enriched tubulin at the microtubule plus end, the mutant, which loses the ability to discriminate between these lattices, doesn't have that preference and is linear all the way to the very end of the microtubule. Further, we can see that it remains in a prolonged attached state to the microtubule tip. So here in these dynamic assays, we're looking at microtubules growing and shrinking. And what we find is the KIF1A wild type is likely to reach the end of the microtubule and then detach when it reaches this GTP-rich region. In contrast, the mutant is likely to stay in a prolonged attached state. So that's all in vitro. What happens if you put this mutant KIF1A back into neurons and ask about the delivery of synaptic vesicle precursors to presynaptic sites? Here you can see the frequency of enriched frequency of pausing that we see for the wild type at presynaptic sites is significantly blunted when we are expressing the mutant KIF1A. And we can show that that blunting leads to decreased retention. That's specific for synaptic sites reached in the anterograde direction. And as a control, we see no change in trafficking in the retrograde direction. So then we can ask what effect this might have on synaptic function. We've already been introduced by Subojit to the pH fluorine assay for V-glut. When it's fused to V-glut, it's a measure of neuronal activity, synaptic activity and release. And what we can see here is for neurons that are expressing the mutant form, we see a decrease in presynaptic strength, both in terms of the release, smaller release, and also a smaller total pool of synaptic vesicle precursors at these sites. And that's further quantitated here, that we see a decrease in presynaptic area as well as total synaptic vesicle pool size. So that gives us a model. I already introduced you to what we think is going on in the wild type. These highly processive kinesins are trafficking down the microtubule with some low but constant probability. They'll detach when they reach these dynamic plus ends and deliver their contents locally. However, in the case of the mutant KIF1A with the, that is no longer able to sense the difference in the lattice structure, you get more random delivery and you get a decreased delivery of synaptic vesicle precursors to presynaptic sites. And finally, Pedro Guedes-Diaz, who did all this work, stepped back and asked, if our measurements are correct, can we explain the overall turnover of synaptic vesicle proteins at synaptic sites? So he took the known known measurements from the literature about the number of synaptic vesicles at a presynapse and hippocampal neurons and their overall lifetime, and put in the new numbers we've measured for the probability of pausing, which is 30% at any site, and the probability of retention, which is quite low but consistent. And all of this together gives us a rate of replenishment of about 198 vesicles per 30 hours, which is remarkably similar, nice balanced budget, better than any of my lab balanced budgets. But really nicely explains the continual stocking of these presynaptic sites. And Lily already explained the extension of this, that when neuronal activity increases, that's going to give you more dynamic microtubules and give you a higher percentage of pausing. So I'm really excited about the data we heard earlier. And I'm just going to end there, since I'm probably over time. Just have to give credit to where it's due. Mara Olenek did all the work on HOOK1 in my lab, and Pedro Guedes-Diaz single-handedly did all the work on KIF1A with a little bit of help from other lab members. And I also encourage you to go see Chantel Evans' poster, which is up hanging, and that's an entirely different subject, so I won't replicate anything that you've heard. And I thank you for your attention.
guess I have a question then, Erika. So Botox, doesn't it take people from picnics, botulinum toxin, don't they have to spend weeks before they get the refill of the effective vesicles? Do you think 30 hours is really enough?
Oh, we just did this in a model, the hippocampal neuron in vitro, so that's a long way to extrapolate to a live animal or human forehead, so I'll punt on that one.
Who's pointing? Somebody's pointing at someone. Are these dense core vesicles?
No, so dense core vesicles show a similar pattern, but not quite as strict. They will pause preferentially at dynamic microtubules, but when you look in the trafficking in the neurons, it's slightly less blunted. They're only pausing at synaptic sites 1.5 times more than you would anywhere else.
How can you tell the two apart? Like an endosome that's moving some synaptic vesicle precursors from a dense core vesicle that's also moving from the Golgi that contains synaptic vesicle
Yes,
precursors.
that's a good question. We did those studies with distinct markers for each compartment, and we didn't do a mixed study that would really answer your question. So that would be something interesting to look at.
Erika, fantastic. I just wanted to ask you one thing in relation to the affinity of the motor to the GTP tubulin. I mean, if I understood, actually the affinity lowers when it encounters the GTP tubulin-rich lattice. Mm-hmm. So that's very clear in the case of dynamic microtubules. But what about nicked microtubules that we know now can self-repair and add GTP tubulin in it? Do you think that's a way that can be regulated by the cell to make the motor fall off?
Yes. Yes. When I heard Antonina at the Garden Conference, I almost fell off my chair. I thought, this is how we could regulate it.
Exactly.
And I think that's something that you should really look at. It's super exciting.
You're a group of Italians stuck in the door, and there's an open row of seats right up here.
Erika, that's a beautiful story.
Oh, thanks.
And it all depends on the microtubules, right? So everything is mode as microtubules. But there's also these factors that, like liprin, tank, that are basically adapters and help the FedEx, to signal where they have to deliver. How does it fit in your model?
Exactly. This is just a way to regulate it, not the way or the only way. RL8 is also involved in this regulation. And I think what this model is just doing is giving you enhanced pausing and not actually involved in the retention. So I think that those factors are very involved in that. So I'm not trying to say this is it. It's a piece of the puzzle that Lily was going to get to very quickly.
Hi, very interesting. You were talking about how this, for KIF1A, how this relates to the increased pausing. What thought do you have about how or whether this relates to some of the thoughts about post-translational modification and how that affects pausing? And sort of what's your thought on that interaction there?
So the obvious thing that you would think would be that tyrosination would be a player because that's going to be enriched at dynamic plus ends. Fortunately, we have a collaboration with Carson Yankee, and he sent us tyrosinated tubulin versus non-tyrosinated tubulin that we could construct microtubules and see if there's any distinct binding difference. Sorry to sneak an extra slide in, but so in our hands we see that KIF1A has no sensitivity for the state of tyrosination on the lattice, which is very distinct from dynein and dynactin, which does respond to a tyrosinated lattice. But that's one of many different modifications, and we do think that other modifications are going to be involved either directly or indirectly. As Casper pointed out, there's many other factors that could be influenced by the post-translational state of the microtubules, such as MAPS and such, which we think are another level involved here.
Have you also looked at post-translational modification of KIF1A?
Oh, no, we have not. Okay. No, we haven't.
Question was not directly, doesn't relate directly to your talk, but this somehow comes to mind thinking about your talk. What prevents the proximal varicosities to be more loaded with vesicle as opposed to the distal one? What is the mechanism that balance the number of vesicle in individual varicosities?
I think that's a great question. I have two answers, entirely speculative, because we know that doesn't happen. We know that you do continue to get delivery. So part of it, I think, is the lowness of these numbers. So pausing is 30%, but retention is down at 1%. So that's going to mean that any given one, it's just going to be a very low frequency of events. And the second, I think we heard really elegantly, that the activity of each individual synapse may then regulate the degree of dynamic microtubules there, which would then further influence these numbers up or down. So I think we're in the beginning stages of understanding this. And again, to speak to Casper's point, I think this is just a way to get the vesicle to pause or to fall off its track, and then other factors will be required to locally enrich it and retain it.
Franck, that's
A very naive question from a non-microtubule person, probably the only one in the room, but how do you relate the two parts of the talk? How is dynein processivity affected by this very phenomenon? Or if, and if it's not, why not?
No, I wasn't trying to relate the two. I was trying to give you two different phases of axonal transport. Just that sometimes it's highly throughput, and it's an expressway. You load the growth factors distally, and you want to get them to the soma as fast as possible. And other times you don't. You have a totally opposite question of local delivery.
So my question is, why would long-range, fast dynein mitosan-directed motors not be affected by the structure of the tube and dynamics of the tube?
Oh.
Can you repeat the question?
Oh, okay. So the question is, why is long-range dynein not affected by the nature of the tubulin here? Actually, it's positively affected because we showed previously that there's EB1, CLIP170, promotion of dynein dynactin binding to the microtubule plus N. So when dynein runs into those loaded plus Ns, they're going to bind more efficiently and then go back to the cell faster. So they're moving along. They might dissociate from 1 minus N. They'll diffuse until they see this enriched plus N with lots of tyrosinated residues and lots of EBs, and then they're going to bind and zoom that way. So in a way, it enhances the quicker retrograde flow.
Because your initial model for dynein initiation was that it happens very distally.
Yes, yes.
Now you're saying that basically along the axon, which makes thousands of passants bouton, each bouton is actually an accelerator?
Yeah, it's an on-ramp, that if you fill off, it's, yes, exactly.
But also because you used the DRG before.
Yes. Yes, yeah. Wow, you guys read my papers.
Use the microphones.
We do.
All right, Dr. Shackman.
How does a non-retained vesicle get back into the mechanism?
Oh, so it's going to have a rebind, it'll diffuse and rebind. So none of these are absolutes, they're just probabilities. So I think if it falls off, it's going to still have a pretty high chance of rebinding to microtubules. Just like this diagram sketches, we don't think, I don't know, maybe Lily and Francesca have a different interpretation. We don't think that all microtubules end there. There's just an enrichment of ends there. And some, as the middle line is suggesting, some will never even see those dynamic ends. The KIF1A is a highly processive motor, so if it's on a microtubule that doesn't happen to end there, it's going to go roaring right through.
Comments? Lily? You want to say anything? No? Okay, anybody else? All right, Erika, thank you very much.
Thanks. So I was a former postdoc in Franck's lab, and now I've moved on to do a second postdoctoral stint in Dr. Beth Stevens' lab at Boston Children's. So today I want to give a good chunk of my time will be used to talk about the work I did in close collaboration with a colleague, a PhD student, Annie Lee. So both of us worked on this project, and then I'll take a few minutes to talk about what I want to do next and get your feedback on the new avenues I'm trying to explore. So thank you for having me. Sorry, this is also a non-microtubule talk. I apologize. But we're going to talk about a favorite organelle in Franck's lab, which is the mitochondria, and how and how dysfunction in mitochondria can lead to neurodegeneration, particularly in Alzheimer's disease. So to start with, I'm not giving great introductions about Alzheimer's disease, but I would like to talk about the key protein amyloid beta, which is one of the pathological hallmarks in AD. It is the constituent of all the extracellular plaques that are found in AD patient brains. And amyloid beta is a really small protein that's
formed by several proteolytic cleavages of a longer protein called amyloid precursor protein. And once it's formed, it's a highly disordered protein. So it starts to aggregate and forms lower order oligomers and further leads to formation of these higher, large plaques in the brain. So what's been known in the field for at least over 10 years now is that lower order oligomers or soluble oligomers of amyloid beta are very toxic to synapses. So if you incubate, this is just a slice culture where you incubate them with either monomers of amyloid beta or with these soluble oligomers. So this paper shows really clearly, this is from Bernardo Sabatini and Dennis Selko's lab. They show that monomers are not toxic for synapses, whereas oligomers incubated over a period of time leads to progressive loss of synapses. So in Franck's lab, we were interested in understanding why this happens and what are the signaling mechanisms that lead to this. But before that, I think this is a great slide that I picked from a review by Morgan Sheng, almost like published several years ago. But I feel like this is a nice slide to represent all the pathogenic mechanisms that has been studied in the field so far, so which kind of unifies the various problems in the cell that can lead to loss of synapses and neurodegeneration. So, but then it also highlights the other key player in Alzheimer's disease, which is the tau protein, and we all know we all know what this does in normal neurons, as well as when they are hyperphosphorylated, it leads to neurodegeneration, particularly in AD. So what's also really nice about this slide is that it seems like there is a lot of unifying hypothesis for generation of amyloid beta and how this can impact on synapses, but also independent mechanisms of like calcium dysregulation or dysregulation of autophagy that can implicate in the end, implicate in neurodegeneration. So I feel like this is a good slide to start with, because towards the end I want to convince that we've added more knowledge to this and a unifying mechanism. And I'll leave it there for now. The last slide is exciting. So to start with, a couple of years ago in Franck's lab, we had identified a really cool pathway that seems to connect, that to my knowledge is the first pathway that connects amyloid beta with tau. So this pathway is basically the CAMKK2 AMPK pathway, which seems to be overactivated upon amyloid beta treatment, and is one of the pathways that leads to synaptic loss in AD. And how was this shown? So, if you genetically ablate or like get rid of the AMPK kinase, either in vitro in primary cortical neurons, or if you overexpress dominant negative forms of CAMKK2 or AMPK in vivo using sparse electroporation in the either wild type or an APP mouse model, what we see is that not having these kinases can completely block synaptic loss. So it seems like amyloid beta has a direct effect on activation of CAMKK2 and AMPK, and this leads to synaptotoxicity. But the next question that we were interested in was the underlying cellular changes that happen and that can lead to synaptotoxicity. And I think this is the best meeting where I can talk about cellular changes and discuss and give you new ideas about what we want to explore. So to begin with, a very brief introduction about AMPK. I really don't know this much about AMPK, but it's definitely a very crucial kinase. It's a central metabolic sensor. It does a lot of things in glucose metabolism, lipid metabolism, protein metabolism. But today for this talk, I think this arm, the autophagy and mitochondrial homeostasis arm of AMPK is something I'll focus on. And I'll give you reasons why I want to focus in this arm of AMPK regulation. And also 2 years ago, there was a paper that was published where they show that AMPK regulates mitochondrial fission in response to energy stress. So this happens during stressful conditions and not like in basal conditions. So why am I focusing on mitochondrial homeostasis? So I think a good justification for this is this slide, which is a review article that was recently published by the PhD student I worked with. So neurons are really beautiful cells which are very highly compartmentalized. And if you look at the axonal mitochondria and compare them with the dendritic mitochondria are really giants, and they are like 20 to 30 times larger than axonal mitochondria. And this degree of compartmentalization is like a fine balance between fission fusion. And this, again, is another paper that's just recently come out, which I just got to know from Franck. One of our colleagues has published that a protein called the mitochondrial fission factor regulates this fine balance of compartmentalization. So why am I talking about this? I think I'm talking about this because the changes that we see, the A-beta mediated changes that we are interested in is synaptotoxicity. And if you look at the dendritic compartment, it's just full of mitochondria. So the obvious question that we wanted to ask was, does A-beta have an effect on dendritic mitochondria? And that was the first question that we asked. So for this, we've used matured primary cortical neurons in culture, and we treat them with oligomeric A-beta, synthetic oligomeric A-beta. And if you look at this, this is a time course of A-beta treatment, and I'm showing here just synaptic loss over a period of time. But the same neurons are transfected with a mitochondrial reporter, and what we notice is that over the same period of time where the synaptic loss occurs, there's also profound changes in mitochondrial morphology. And also there's a profound loss of mitochondria over 24 hours of A-beta treatment. So the other question that we have is that since we found a pathway that regulates synaptotoxicity, which is mediated by AMPK, does blocking AMPK have an effect on mitochondrial morphology? So for this, we've used neurons from AMPK alpha 1 and 2 floxed mice, and we reconstitute these neurons in vitro plus minus Cre, and treat the primary neurons with and without A-beta. So what we observe here is that when you do not have AMPK alpha 1 or alpha 2, we actually don't see any changes in either mitochondrial morphology. So all of the mitochondria exist intact in the dendrites, and the synapses are also normal upon A-beta treatment. So there seems to be AMPK plays a profound role in, not having AMPK plays a role in blocking both synaptotoxicity as well as mitochondrial toxicity. So this is all in vitro. So what happens in vivo in the AD brain? And that's also a key question that we wanted to explore. And for this, we used a technique that Franck's lab has used over many years now, where we can sparsely label neurons in vivo using in utero electroporation. And in this technique, what we've done is we've just labeled neurons with a cell filler and a mitochondrial reporter. And we look at wild-type brain as well as the J20 mouse model, which overexpresses APP and therefore will give us answers to amyloid-mediated effects on neurons, on the hippocampal neurons. So what we first see is, apart from the AD brain, I think I'll just talk about the wild-type brain first, because there was an interesting phenotype that was observed in just wild-type hippocampal neurons. So wild-type hippocampal neurons seem to have a subdendritic compartmentalization of mitochondria. So they have these, this is more appreciable in the next slide. I think it's too small to appreciate it here, but it seems like in cortical neurons, mitochondria are really long throughout the dendritic arbor, whereas in hippocampal neurons, they are small and punctate in the basal region, and they start becoming really long towards the distal apical dendrites. And that is exactly what this slide shows. So in the basal and oblique region, the mitochondria are small, whereas in the distal apical dendrites, they are really long. And what is so special about these distal apical dendrites in context to Alzheimer's disease? So when we were trying to look for synapse changes in the AD mice model, so what we found was that this is the first region where a profound loss of synapses occurs at early stages of disease at 3 months of age. And beautifully in the same region, we also see profound changes in mitochondrial structure in terms of loss of mitochondria. So this was really promising because it seems like there is a physiological mechanism of mitochondrial loss and its implication in Alzheimer's disease. And these are the quantifications for the in vivo analysis, where we see profound loss of synapses at the distal apical region as well as mitochondrial density. Some amount, a trend in the basal region, but not so much with the synaptic density. So it seems like the distal apical region are really vulnerable, the earliest region that's vulnerable in the disease. And the vulnerability is caused by changes in mitochondrial structure. So to explore further, so we see a significant reduction in mitochondrial density. So what is the mechanism by which this can happen? And can mitochondria just like, how do they disappear? So for this, we used our in vitro model where we wanted to test if mitophagy has any role in this process. And mitophagy has been studied in neurons, not to a great extent, but I think the current running hypothesis is that mitophagy can occur in distal axons in a local degradation manner. So because axons can run to long distances, so it seems more energy efficient to degrade these mitochondria in by local degradation rather than transporting them all the way back to the soma. So since we were seeing changes in mitochondria in dendrites, and dendritic mitophagy has not been very well studied at all, so we wanted to know if what happens in the dendrites, how are these mitochondria disappearing? So for this, we just used primary cortical neurons, where we labeled mitochondria with mito-BFP, and we label lysosomes with LAMP1 and autophagosomes with RFPLC3. And we treat them either with DMSO or A-beta-42 and track the dynamics over 14 hours. So what we see in control conditions is that the mitochondria in the dendrites are just about like sitting there wiggling around. And then you have nice movement of autophagic structures in and around in the dendrites. And then once we start treating these neurons with A-beta, nothing happens for a while, and then after a period of time, there's this sudden influx of lysosomal compartments like LAMP1 as well as RFPLC3, and they start pausing at mitochondria, near mitochondria, I would say. So to quantify this dynamics, what we did, the next slide will just represent how we tried to quantify mitophagy using this in vitro system. So basically, it's a very crowded slide, so I'll go over this a bit slowly. So this is just control condition with 0 and 14 hours, tracked from 0 to 14 hours, and you see LAMP1 and LC3 structures staying near the mitochondria over the period of 14 hours, and that is also represented in the kymograph here, where they stick around for 14 hours. And in those regions, so basically we picked, and this, I'm jumping too fast. So this is the A-beta-42 condition where again we track for 14 hours, and you see that there are some de novo structures that start appearing during the course of A-beta treatment of both lysosomes and autophagosomes. And the way we try to quantify this, this is to look for regions that either have LAMP1 and LC3 or lysosomes, autophagosomes, and regions that do not have these structures, and look for changes in mitochondrial intensity over a period of time. And what we observe is that only upon A-beta treatment, where regions where the LAMP1 and LC3 begin to appear, you start seeing a decrease in mitochondrial intensity over this 14 hours time point. So it seems like there is, first of all, like since we start seeing an influx of lysosomes towards the mitochondria, we think it's local degradation in the dendrites, and probably this is probably the first evidence of local degradation in dendritic mitochondria in context of dendritic mitophagy. So to further delve into the signaling pathways that can coherently regulate synaptic loss and mitochondrial changes, again, I've taken a candidate-based approach, focusing on the mitochondrial homeostasis arm of AMPK, where AMPK is very well studied in non-neuronal cells to have a positive, to regulate autophagy positively by its substrate ALK1, by acting on its substrate ALK1. And like I said, 2 years ago, it was discovered that AMPK can also induce mitochondrial fission upon energy stress by phosphorylating a protein called mitochondrial fission factor. So looking at these two, we started by looking at these two proteins to begin with. And one reason why we think this made logical sense is that a running hypothesis in the mitophagy field is that in order to get rid of dysfunctional organelles, mitochondrial fission is kind of a prerequisite to sequester dysfunctional organelles into the autophagy compartments. So we think that since AMPK coherently regulates these two fundamental mechanisms in mitophagy, this seems to be a logical manner in which we wanted to test this. So to first start with, we knocked down the mitochondrial fission factor, or we downregulated mitochondrial fission in primary neurons, and we treat these with and without A beta, and look for changes in both synapses as well as mitochondrial morphology. So what we see is that if you do not have mitochondrial fission factor, we can rescue synaptic loss, as well as, we can block synaptic loss as well as the changes in mitochondria in these neurons. So mitochondrial fission, therefore, is key to cause this pathogenic cascade. But another interesting thing that we tried was to knock out the ULK kinase. So basically, in non-neuronal cells, it's found that ULK1 is the substrate of AMPK, whereas in neurons, it seems like ULK1 does not play a role, or rather, probably it's just very low, expressed at low levels. So we've done a panel where we tested the ULK1 and ULK2, and what we found is that it's ULK2 lies downstream of AMPK in neurons. And we've used ULK2 floxed mice, which are reconstituted minus plus Cre, and again, the question here is, what happens when you inhibit mitophagy and you look at these A-beta treated conditions? It was really interesting to find that, so if you look at this panel here with A-beta, you have a loss of mitochondria, and then when you inhibit mitophagy, you seem to still have mitochondrial fission happening in this condition, but there is a whole load of small mitochondria present in the dendrite, and that was sufficient to rescue synaptic loss, or sufficient to prevent synaptic loss. So I feel like this is probably one of the, I can't say best evidence, but maybe I can here in this audience, probably a good evidence for a causal relationship between mitochondrial biomass and synaptic maintenance. And one possible reason we think this could happen is the crucial role mitochondria play in calcium buffering properties in neurons. Yeah. So these are the quantifications for both these conditions, for the MFF knockdown and ULK2, where we show that mitochondrial density is blocked and synaptic density is blocked, and likewise for the ULK knockdown, mitochondrial density, synaptic density is blocked, whereas the length still is significantly less. So, yeah. And then as an extension of this study, we also want to show that this is indeed AMPK dependent, and for which we had a collaboration with Ruben Shaw's group, who was the group that originally identified the signaling pathways between AMPK and MFF and ULK. And they've helped us to validate that ULK2 is also a bona fide AMPK substrate. We predicted a few phosphorylation sites on ULK, which we mutated, and by biochemical analysis, we were able to show that this is AMPK dependent. And likewise, we were also able to show that MFF is phosphorylated in neurons by A-beta treatment, and it's also, this is just a control in neuro-2A cells where we treat with metformin, that's an AMPK activator, and show that that happens even in other cell lines. So the reason we did this experiment was then to generate phosphodeficient mutants of MFF and ULK. So basically, we made alanine mutations of all these phosphorylation sites, and then we want to test these in primary neurons. And for this, we took a gene replacement strategy where we basically knocked down endogenous mitochondrial fission factor in primary neurons and reconstitute these with either wild-type MFF or a phosphodeficient MFF. And what we see is that only under conditions of wild-type MFF, A-beta is able to cause synaptotoxicity as well as an effect on mitochondria, whereas when you have the phosphodeficient mutant, we do not see this effect of A-beta on mitochondrial morphology or synaptic loss, which clearly shows that AMPK is upstream of MFF in this whole synaptotoxicity cascade. And likewise for ULK, we took a gene replacement strategy where we can put back the wild-type form of ULK. And ULK2 is also a kinase, so this is like a whole load of kinases there that are activating one after the other. So for that, we also generated a kinase inactive version of ULK, as well as the phosphodeficient mutant of ULK. And basically, we reconstitute these neurons with these various versions of ULK. And then we ask what's their effect on A-beta treatment. And what we observe is that only under conditions of wild-type ULK, we see both reduction in mitochondrial density as well as synapse density, but not in conditions where we express kinase inactive or phosphodeficient mutant of ULK. So once again, highlighting the fact that AMPK is upstream of this cascade. And there is a signaling between AMPK and ULK2 in synaptotoxicity. So, so going to the grand scheme of the model, where we're trying to claim that, so basically we think that in the healthy brain or in healthy hippocampal neurons, you have influx of calcium, and mitochondria are really crucial for buffering calcium in these dendrites. Whereas in the Alzheimer's brain, what we've observed in vivo is this profound change in mitochondria, profound loss of mitochondria, I should say, in these distal regions. And the implication of loss of mitochondria, I think, is that it's key function for calcium buffering. So what we think is that upon A-beta treatment or in the presence of A-beta, there's an influx of calcium, huge influx of calcium, which activates the CAMKK-AMPK pathway, which coherently overactivates or like coordinates mitochondrial fission and mitophagy and leads to loss of mitochondria. And since we have lesser mitochondria in these dendrites, there's a whole load of cytosolic calcium that is accumulating in the dendrites, and this feeds back into this calcium-dependent pathway. So we think this is some sort of like a vicious cycle, and we think it's truly a calcium-dependent synaptic loss, and local changes in calcium can influence loss of synapses. So going back to the first slide that or the second slide that I had, where I spoke about all these different mechanisms that have been worked out over the past 20 years in Alzheimer's disease, I think we can confidently add this mitochondria hypothesis now, claiming that changes in mitochondrial architecture or loss of mitochondria can contribute to early synaptic loss in Alzheimer's disease and further neurodegeneration. But, 2 minutes. Okay, sorry. And and we think that the CAMKK2 AMPK is coordinating most of these things that we are talking about here. So like A-beta has an effect on CAMKK2, and AMPK also has implications in increased autophagy and on tau. But just taking really 1 minute to describe what I want to do next is I've moved on to Dr. Beth Stevens' lab, where they are focusing on the microglia hypothesis, which is a fairly new hypothesis that's been described in AD, where they show complement activation in Alzheimer's models as well as tau models, amyloid models as well as tau models, and they claim that complement can tag synapses at a really early stage and leads to microglia-mediated elimination. So what I'm trying to ask here is there a crosstalk between neurons and microglia and during early synaptic loss in Alzheimer's disease? And I think this is the next step which I want to take in this project. So I thank you all for your attention, and this is the most important slide. I thank Franck for being a great mentor and the people we closely worked with and everybody else, collaborators. So thanks a lot. Yeah.
Very nice work. Have you tried to counteract the fission, excessive fission, by overexpressing, for instance, something like mitofusins to see if you then prevent mitophagy?
No, we have not tried mitofusins, and I'm not entirely sure if overexpression of mitofusin is even healthy for neurons. So, but yeah, we've not tried.
And ATP levels, have you looked into this as well?
Not yet. We haven't looked at the function of mitochondria, and yeah. So we don't know what happens to. Yeah, so when we knock down MFF, there is no mitophagy. So under basal conditions at least, yeah.
Very nice. I was wondering if you characterize the mitophagy that you're seeing. Is it PARC independent? Is it involve optineurin and the other receptors?
Yeah, so we started a little bit on optineurin, but I think it kind of like stopped because it was really tough to track. Optineurin dynamics as well. But yeah, no, we have not gone into like pink parkin as yet. Yeah.
say Dottore Maria Concetta Minacci, and that takes the time that it takes to watch the back.
Thank you for your talk. So I have a question, whether you look at the mTOR level, because the AMP kinase has a crosstalk with the mTOR. So whenever there is energy deficiency for several reasons, so the AMP kinase increase and the mTOR decrease, and so maybe the synaptic loss you see, and also there is also an increase of autophagy is due to the decrease of mTOR level, for example.
Yeah, I mean, it's totally possible. I think we tried initially to do some biochemistry with these primary neurons, and no, we kind of failed.
Yeah, because
I think
it's
it's
very
just
interesting, this mTOR
Yeah.
and crosstalk.
Yeah, so we didn't clearly look at mTOR, but we do see activation of AMPK, and I'm sure that will have implications on mTOR signaling
Yeah,
in
because
terms
it's
of
also
autophagy.
involved in autophagy.
Yeah,
Yeah.
yeah.
Are you biting off pieces of sick mitochondria like the yeast people like to think?
What's that, sorry?
When you're doing the fission, are you doing this in order to bite off the pieces of, are you swallowing entire mitochondria, or are you only swallowing the sick parts?
We think it's the sick parts. Yeah.
Anybody else?
But yeah, we have no clear evidence, right?
Okay, Chandana, thank you very much.
Thank you.
Do you want to say something about this?